what do we know about resistance? In scrapie, we know that those amino acids that they've identified that contribute some level of resistance can completely prevent sheep with those markers from getting scrapie altogether. In deer, we're trying to figure out whether that's the case or not. But, you know, some of you have already seen this, but, you know, what we have up here is a water-resistant watch. So what does it mean when you have a water-resistant watch? Does that mean that it's waterproof? So you guys can answer questions. Does it mean that it's waterproof? What does it typically mean if you have a water-resistant watch? Yeah, so 3 feet, or some of them are rated to 50 feet or 50 meters. You know, the level of resistance really depends on how much it's exposed to. If you have a water-resistant phone, for example, that doesn't mean you can go swimming with it. It means that if it gets splashed with water while you're at the pool, you know, you can wipe it off and chances are it'll be okay. What we're trying to identify here is exactly how much CWD can some of these animals with these markers be exposed to before they develop CWD. So that's the question right now. So if you hear people selling straws of semen from deer that they're saying are CWD immune, don't buy that. We don't know that yet. We know they have some level of resistance. And you know, one of our objectives for the study that we're proposing right now is to figure out exactly how resistant they are. If they're not completely resistant, can we figure out ways to help manage CWD? You know, given that maybe the disease, we can extend the disease from 2 years to 6 or 8 years. And so, you know, a lot of this we're still trying to figure out. But we'll hear arguments from people saying that this doesn't mean that these animals are resistant. It just means they're less susceptible. When I hear those, you know, less susceptible, resistant, those words mean the same thing to me. So a term I heard somebody use a couple weeks ago is that maybe they're more CWD tolerant, where they can be exposed to more of it and are less likely to develop CWD given that level of exposure. But however you want to term it, less susceptible, somewhat resistant. We're still trying to figure out exactly what that all means. In general, we know that animals with these resistant markers tend to have longer incubation periods. So, you know, they're exposed and it takes them 4 to 6 years to develop CWD as opposed to 2 years with some of the other markers. We don't know anything about when they start shedding, when we might be able to identify them through rectal biopsy testing, for example. We just know that it takes them much longer to get sick than it would otherwise. So going back to this cartoon that I have up here with the Legos, a lot of work has been done on prion resistance in different species. So work has been done in humans, work has been done in goats, in sheep, like I mentioned with scrapie, in deer, and in elk. And most of the markers for resistance fall within that sort of Lego portion of the protein of the gene. In deer, up until about 12 months ago, there were basically 4 different markers that had been identified, and no one really knew exactly how resistant some of them were. Most of the talk had been about 96 GRS, and some of you may have already heard those markers being thrown around. That's because 96G is the most common marker that's out there. It also happens to be the most susceptible. 96S is the second most common, and it's kind of marginally resistant. But there's several other markers, 95H, 226K, and just within the past 12 months between Dennis and myself, we've identified 3 or 4 new markers that had never been reported before, and those may have some importance as well. So it's something that we need to kind of look more into. This is playing out exactly how scrapie resistance in sheep played out. You know, once a few markers were identified, you know, a huge net was cast trying to find other markers, and eventually a solid plan for breeding genetic resistance in sheep was developed. And over the past 15 years, that breeding plan has been pretty successful in eliminating scrapie from the United States, from Europe and Great Britain, So, in most cases, the number of scrapie cases in those countries has been decreased by 95% or more just through breeding genetic resistance. So I think that's our gold standard. We're hoping to have a resistance level like that imparted by some of these genes, but we need to do the work to kind of figure that out. So just a little bit more of a background on the sheep scrapie markers. A few of the positions have been identified as very important for disease resistance in sheep scrapie. 136, so sheep can have amino acid V or A, and don't get too caught up in where these positions are, what the letters mean. They're basically just differences, just like you could have differences on your key that make it suitable for fitting one lock or another. 154, H or R, 171, Q or R, and in general, animals that have a V at 136, an R or an H at 154, and a Q at 171 are extremely susceptible to scrapie. And then animals with what we call ARR, so an A at 136, an R at 154, and an R at 171. Those are the animals that are extremely resistant to scrapie. And over time, what they've figured out is that if a sheep that has that VRQ combination of markers is exposed to scrapie, they'll usually develop disease within about 2 years. The ARQ animals or AHQ animals may go a little bit longer, so they may go about 3 years after exposure before they start showing signs of scrapie. And then the ARR animals, so animals with an ARR from mom and an ARR from dad, when they get those markers, the disease process essentially goes longer than a sheep is capable of living. So, they're exposed, and if sheep live to be 20 years old, they may develop scrapie, but most sheep don't live until they're 20. Most of them will be dead by the time they're 12. So, practically, these animals, sheep with these particular markers, are considered completely resistant to scrapie, and they won't ever develop in their natural lifespan. So tables were developed. A lot of work was being done on identifying exactly how resistant some of these markers were in sheep. And so just comparing the top and the bottom lines of that table there, VRQ animals, so animals that got a VRQ marker from mom and a VRQ marker from dad, those essentially have no resistance to scrapie. Animals with an ARR marker from mom and an AR marker from dad, those animals are considered 13 times more resistant to scrapie than the VRQ animals. So this is all work that's been done essentially with the help of sheep farmers who had scrapie identified, and through the work of the USDA, Canadian Food Inspection Agency, and agricultural agencies in Europe. They had to go through all the depopulations and sort out exactly how resistant each of these different markers would be. So what do we know about deer? So like I mentioned, up until about 12 months ago, we knew about 4 different markers. We have position 95, which is commonly a Q, 96, which is commonly a G, 116G, which is commonly an A, and 226, which is commonly a Q. These are all in the allele that we're calling 96G. And again, the 96G allele is considered the most susceptible to CWD. We have what's called the 96S, so it's identical to the 96G allele. The only difference is at position 96, there's an S instead of a G. And then the other markers that we know about, 116G, essentially identical to the 96G. The only difference is there's a G at position 196, 95H, identical to the 96G. The only difference is an H at 95 and 226. And you kind of get the picture. These are just single amino acid differences in that protein, single building blocks that are different, that are different enough that prevent that lock from fitting that key as well as it would have, and impart some level of resistance to CWD. So one of the problems that we've run into with testing is, because 96G and 96S are the two most common alleles, the USDA started focusing on those right away. And so they developed a test that only looked at position 96. So if you only look at position 96, the only difference that you're going to see is either a G or an S. So what does a 116G look like at position 96? So it looks like it's a 96G. If you look at 95H, what does it look like at position 96? Same thing. So it looks like it's a 96G. So, the testing that has been developed by the USDA at this point and that's being implemented at this point kind of ignores all these other markers that may provide some level of resistance. So they're kind of missing, a good portion of the picture, which can be a good and a bad thing depending on how you're looking at it. And we'll talk a little bit about that later. So all of that work that's kind of gotten some insight into resistance or reduced susceptibility in deer has been done in wild deer populations. So some of those studies were done right here in Wisconsin. Most of that work was done 10 or 15 years ago or more in some of those counties that had high levels of CWD, Dane and Iowa County. But not a lot of work had been done in the farm deer industry. Not a lot of work has been done in the past 5 or 10 years on looking at these markers and trying to identify how resistant they are. So what we've done is two things. We've gone into all the herd depopulations that we could in both the US and Canada over the past 10 years or so. We've got numbers totaling a little bit over 2,000 deer that we've looked at now. And about a third of them had CWD. The other thing is we looked at stage of disease because up until this point, the only thing that they'd been looking at was are the disease positive, or are the deer positive or negative for CWD? But it's a little bit more, it's a little bit more detailed than that because we can figure out exactly what stage of disease the animals are in. Are they in an early stage of disease or are they in a late stage of disease? So going back to the Alzheimer's analogy, most of you probably know of somebody who's had Alzheimer's, and very early on in disease, the signs are subtle. They may forget where they put their phone or where they put their keys. And then over the course of a few years, it starts to become more significant where they don't remember somebody's name. They don't remember how to get to the grocery store. And eventually they wind up getting lost. And in Phoenix, people get lost in the desert, and we have what's called silver alerts. And we have to be on the lookout for some car that's driven off and nobody knows where they went. So disease stage is important, not just whether the animals are positive or negative. So this is kind of the raw data from what we collected over the past 10 years from deer depopulations. I know it's hard for some of you to see, but towards the left are the animals that are what we call homozygous for 96G, so they're 96GG animals. Those animals are as susceptible as it gets. The color of the bars in this bar graph indicate what stage of disease they're in. So what I hope you can see is that the 96GG on the far left, a lot of those animals are in later stages of disease. So the red is sort of a late stage disease and the green is an early stage of disease. So those animals on the left, the 96GGs, tend to be in much later stages of disease when they're identified as positive. And then we can just go down the chart to the right, and the likelihood of identifying, for example, a 96SS animal that's positive is a little bit less than half of the likelihood of identifying a GG animal as positive. What I want you to also see is that all of the animals that are 96S, so the 96SS animals that we've identified as positive, they're all in the very earliest stages of disease. So if you're familiar with how the testing is done, we look at the lymph node and we look at the brain. All of the 96SS animals that have been positive that I've seen that have been reported are all lymph node positive. So, the pathology hasn't reached the brain. The agent hasn't reached the brain yet to any significant extent that they can identify it in the brain yet. So they're all in very early stages of disease, and that kind of, that's kind of the point where, they're forgetting where they put their keys or where they left their wallet, that sort of thing. Some of these markers, 226K, 95H, 116G, they're very rare, so they're very difficult to find paired together as homozygous animals. So it's rare to find a 116, 116G animal. It's rare to find a 226K, 226K animal. So we don't really know how resistant they might be because we didn't see any of them, or we didn't see very many of them in our depopulated herds. But chances are, for example, just having one 226K marker, so the 96G 226K animals, they're about half as likely as a 96GG, and again, they're all lymph node positive only. So the hope is if they have two of those markers, they may be completely resistant. They may be 10 times as resistant, or 1/10 as susceptible as a 96GG animal. These are the sort of things that we need to figure out going forward. But the hope is that some of these markers provide a very high level of resistance to this disease. So I kind of touched on how rare they are. 96GG animals make up about half of the deer in the farm surveyed population. 96GS and 96SS make up about another 10%. So 60% of the animals out there are either GG, GS, or SS. And then the rest of them, depending on whether we're talking about Canada or the United States, are anywhere between 1% and 10% of the population. 116G I haven't talked about too much. We've only ever seen animals with those markers in Canadian farm deer herds. We know that they are in wild deer herds because there was a report out of Wisconsin about 15 years ago that identified some animals with those markers in Wisconsin in the wild deer. But so far, I've only seen them in Canadian farm deer. 226K has a similar story where we only see that 226K marker here in farm deer in the United States. Haven't seen it yet in any of the Canadian deer herds. So those few guys that are still farming white-tailed deer up in Canada are very keen on getting some animals with the 226K marker because it appears to be one of the more resistant markers. More data needs to be collected on some of these animals with double markers because, like I said, they're pretty rare. So we don't see very many of them in the depopulated herds. We can kind of estimate how resistant they might be, but because they're so rare, that estimate range is pretty wide. So, the hope is, going back to that image that I showed before of Scrapie, we have 96GG animals. They're exposed. Within 2 years, they're developing clinical CWD. They're losing weight. They're kind of having severe behavioral abnormalities, and just like people with Alzheimer's, they tend to get pneumonia because they're not able to swallow correctly. These are the animals that are getting gored or may die during fawning just because they're not up to the stresses of a regular routine life for a white-tailed deer. 96SS animals, when they're exposed, they may go out a little bit longer. They may go 3 or 4 years before they start showing these clinical signs. The hope is that animals with double markers for the 226K or 95H might extend beyond 5 or even 8 years before they start showing clinical signs. So you guys who are farming deer know that that's a number that you can work with. If you can keep animals alive for 8 years before they develop disease, that helps people like Greg Fleece, who's harvesting bucks at 3 years of age, and it certainly helps those of you who are trying to have some sort of turnover. It's certainly better than having animals die in a year or two, right? One of the questions is, if we're doing live animal testing, one of our easiest approaches to live animal testing is a rectal biopsy. So we can collect a piece of tissue from the rectal area and run a couple different tests on that. One of the problems is that obviously if they're moving much quicker through the disease process, we can find that evidence of infection pretty early in 96GG animals, whereas you know, it might take a few years longer for it to show up there in that rectal tissue in animals with some of these markers. So the disease process is longer. It takes a little bit longer to identify them as infected, and that could have some implications when it comes to animal movement or quarantine or that sort of thing. The other big question is, when do they start shedding? So, you know, if we can extend the disease process out, but they all start to shed, you know, 6 months after they're exposed, that really doesn't help us out much. So what I hope is going to play out is that these animals with these markers, it takes them longer to get sick, and it also takes them longer to start shedding. Because if they're not shedding until 4 or 5 years after they're exposed, again, that's something you can manage, because both in the farmed and the wild deer herds, your average age of a buck is 3 years or less, right? The does are the ones that tend to live a little bit longer, but we can sort of look at management strategies and maybe have a higher turnover rate of does, so that we're not turning over animals, or so that we are turning over animals before they may start shedding. Is there a question in the back? Good question. So right now, the evidence is kind of mixed. There is some evidence, and so it depends on who you ask. If you ask people on the wildlife side of things, they'd say that they all start shedding right away. There is some evidence for that, but I question that the approaches that were used to come up with that evidence. There's some evidence that's going to be coming out soon that does suggest that they're not shedding, at least in the only markers that have been looked at are 96 GRS, and there's some evidence that animals with that 96S marker, you know, may start shedding 6 or 12 months later than animals that are 96 GG. Does that make sense? So, and I'll talk a little bit more about, you know, the purpose of this study, and that's one of the purposes of the study, is to figure out when they start shedding. But the hope is that it does take them longer before they start shedding. Yeah, so that's the question that's on our mind right now. So one thing we've done over the past year is we've looked at, you know, where these different markers are across the United States. It's pretty similar across all the different populations that we've looked at. Roughly 55% of the farm deer out there are 96 GG. In some states like Texas, the 96S marker is much more common than it is in other states. Pennsylvania and Wisconsin, we've seen a lot of the 95H and the 226K markers. You know, so things vary slightly, but generally, most of the animals are 96 GG. And then, you know, depending on what state we're looking at, and it's really a roll of the dice, because, you know, some of you are probably aware, you know, you pick deer to use as sires based on how much it costs for a straw of semen, or how much the sire costs itself. And so, you know, it's really been kind of a roll of the dice how much each of these markers have been injected into a specific herd. For example, you know, some herds we don't see it at all. Some herds where there's been a lot of line breeding and they happen to select they were lucky enough to select the sire with the 226K. These markers are pretty common. So it really depends on who you decided on as a sire, and how lucky you were as far as how common these markers might be in your herd. So where do we go from here? So there's a couple of things that we've been working on, a couple of things that we hope to start working on. I think it's important to keep genotyping animals, whether they're in a healthy herd or in a herd that's about to be depopulated. So, you know, I hate to say it, but chances are somebody in this room in the next 3 to 5 years is gonna wind up with a positive. If that's the case, and I hope that's not the case, you guys really need to demand that genetic testing is done on the animals so that we can get more information. The information that we have so far has been very useful, but we need information like what pens are each of the animals in, because all the information I have right now, for example, I worked on a depopulation in Iowa a few years ago, and we basically just had, here are 350 deer here, and you know, Of course, all those deer were in different pens, and we would have a lot more useful information if we knew what pens they came out of. But you know, I just want to stress that you guys need to kind of ask for this genetic testing to be done if you wind up in under quarantine and going through depopulation. And until then, I think it's helpful to, you know, if you're interested, and I'm not trying to push this at all. I mean, I'm trying to show you the data so you can make the choice for yourself. But you know, if I were a deer farmer, I would probably be testing my deer for these different genetic markers. If I were just starting out, I'd want to start out with a herd that already had these genetic markers and made it a little bit easier. Because like I said, that 96G marker is very common. And once it's, you know, if you start out with something that's very common, it's a lot harder to get rid of than if you start out with one of these more rare resistant alleles. So there are sequencing services. So Dennis Simpson offers one. North American Deer Registry offers one. I had been running a test in my lab, but just kind of got caught up in too much of this research in order to keep doing it as a contract service. But there are labs out there that can run this testing. They can test hair, semen, blood, you know, antler core, all those sorts of things. And Dennis is going to talk about exactly how to process those samples and send them in. And like I said, if you find yourself in a depopulation event, request that this testing gets done so that we can catch this information. We're also working with one particular farm here in Wisconsin on trying to shift the genetics on that property so that we can reduce the prevalence of CWD and make it easier for him to operate. And I'll talk a little bit about that. This farm has multiple properties. There's one property that has a fairly high prevalence of CWD, looking at 60 to 70% of the animals coming off it are infected this past year. And another property with a low prevalence, so something like 5% of the animals coming off this year have been found to be positive. And we're looking to shift the genetics on that particular operation and seeing how effective we can be at reducing the number of animals turning up positive. One thing that we want to look at is things like fawning rate and fawn health. One of the questions that has been raised is, you know, is there a reason why these animals with these markers are so rare? You know, if they make up less than 10% of the deer out there, is something wrong with them? You know, maybe they don't have high fawning rates, or they have low fawning rates, or they have low birth weight, or the fawns aren't gaining weight appropriately. So we want to kind of pay attention to that to make sure the fawns that have these markers that we're interested in are otherwise healthy and you know, not at risk of higher risk of E. coli or Clostridium or something like that for whatever reason. This is just some data on the farm that we're working with that has CWD on a couple different properties. The numbers are, you know, they're kind of rough numbers. We don't have a lot of 226K 96G animals, but 3 out of 9 of those animals have been found to be infected. A couple of them have been positive in the brain, but by far the most animals coming off of that site, off of those 2 sites, are 96GG, and the prevalence just over the past 2 or 3 years is just a little bit under 50%, and it keeps going up year after year. The GS, you look, the prevalence might look to be a little bit lower in those animals, but you know, those numbers are low enough now that we're not statistically sure how different they might be yet. But it's obviously something that we want to pay close attention to make sure that the way that we're shifting the genetics is appropriate for what the disease is doing on those 2 properties. Just, you know, some of you might have a question as to, if I have a herd of 100 deer, how quickly can I change my herd over to either of these markers? And the answer is pretty quick if you're dedicated. So this particular farm, again, in 2017, close to 90% of the animals had that 96G marker. You know, maybe 50% of them were 96GG, but at least 90% had one 96G marker. In 2018, We started working with them in the fall of 2018 to identify animals with these specific markers so they could try to shift their breeding program over and try to increase the number of animals with these markers. We saw a little bit of an impact, but what I think these numbers don't show is that we went from 50 or 60% of the deer being 96 GG down to 40 or a little bit less than 40 being 96 GG. So more of the animals are having at least one of these resistant markers, either 96S or 226K or 95H. In 2019, we're expecting fawns. Now we've cut down the GG animals considerably. There probably won't be very many 96 GG animals at all. 80% of the animals should have at least one of these markers. And once you identify sires that you want to use for your breeding program and that have these specific markers, it's going to become easier and easier to shift your herd over very quickly. But I anticipate, just another couple of years, and we'll have that 96G allele completely removed from this herd, and we'll be dealing with only less susceptible animals in that herd. So the goal for that project is to see, when we start turning these animals out into some of those into the high prevalence property, can we cut the prevalence down from 60 or 70% down to 5% or less? So that's our goal for that property. So one other important thing that we want to do, because that's a very passive study. We're basically looking, can we shift the genetics in a herd and reduce the prevalence so that farms or ranches that have CWD can continue to operate, given that, in some parts of the state, 50% of the deer in the wild populations have CWD too. But one other thing that we want to do that's very important that, we're trying to get funding for right now is to develop a pen study. And this goes back to the question earlier, to, truly figure out when these animals with these markers start shedding, and when can we identify them as rectal biopsy positive, and ultimately, how long do they live when they have the disease, and is there any impact on the number of animals infected overall with these specific markers? So we'll be looking at shedding and things like saliva and urine and feces and blood. We'll be looking at live animal testing by collecting biopsies, and we'll also be looking at any changes in the prion agent itself to see if there's any sort of adaptation or strain variation that's coming out of these animals with these different markers. I know this is going to be difficult for most of you to see from the back, but basically what we're shooting for are 10 groups of animals made up of 6 animals each. Each of those groups will have different combinations of the markers. We want to have the more susceptible ones in there just so we have kind of a basis to compare back to. We want to sample the animals every 4 months, collect things like rectal biopsies and blood and urine and saliva to see if we can identify it in those samples ahead of time. And then once the animals start showing clinical signs, once they start losing weight or showing behavioral abnormalities, we'll euthanize them and do a full necropsy on them, collect tissues, and get a better understanding of the disease process as a whole, using a couple of different techniques. But obviously we'll be sending in brain and lymph nodes in for testing as well, so that we can see if the pathology there is any different between these animals with different markers. But that's the goal of the study. We have one site that we're very particular about, that has a very good handling facility, has a history of CWD on the site. It was depopulated this year, and the goal is to develop this study on that property and let the animals go out for up to 8 to 10 years, as long as it takes to get answers on how susceptible the resistant markers are, when they start shedding, and when we can identify them as positive. So, so that's our goal, to develop this 10 study where we have fawns that are expected this spring. And the hope is to start that study this fall. And we have a lot of work to do. It's a very ambitious project, I've been told. And so, that's our hope, to get this started this fall. Some questions I've already kind of touched on. Why are resistant animals rare? How does all of this affect trace outs? How does this affect live animal testing? Can we do any better than a 226K or a 95H or a 116G? Are there some genotypes that may be more susceptible to CWD strains? So we don't know much about CWD strains, other than there probably are different strains of CWD. But are some of them more infectious to 96SS animals or what have you? So we want to get more information on that. But to answer the question about why are resistant animals rare, we don't really know. I mean, it may be that they can't do calculus very well, and what does that matter for a farm deer? It may not matter at all. But the question is, why are they so rare? I think it's just because they were fairly recent mutations and they haven't had a lot of time to spread. But in some cases, we've seen, if any of you have been following this, that the 226K seems to be passed down much more commonly than a 96G. So, I think we're just very early on in understanding what these markers, might do as far as the entire animal's health. But that's one thing that we hope to do in the field project that we're working on, is to look at the fawns that have these markers and try to see if they have any sort of negative health outcomes. See if they have problems with weight gain. I don't expect there will be any problems at all in animals with these markers, but we want to be sure. We don't want to try to breed something that could do damage to the industry. That's happened before in the dairy farming industry, where a popular sire had a mutation that caused pretty severe immune dysfunction, and it wasn't identified until decades after that sire had passed away. Although a good proportion of dairy cattle out there had that sire in the pedigree. So we want to make sure that something like that doesn't happen in this industry. How will resistance affect trace outs or live animal testing? Like I said, currently the USDA is only looking at position 96. So if you have a herd full of 226KK animals, at the moment they're going to look like 96GG, because that's all the USDA is looking for. And every time I say that, I know they're listening and they're thinking about it. And the USDA moves a little bit slow, so this may take them a year or two to catch on, but eventually they will. But right now, a 226KK animal would be considered a 96GG, because that's all they're looking at. Like I said, it will take them, because it takes them longer to develop disease. It also takes them longer to show up as positive by for example, rectal biopsy testing. But they're less likely to be infected overall. So, there's some discussion by the USDA whether they should be doing live animal testing to help people get off of quarantine or help them get out of a trace out, for example. And so the USDA is recommending that everybody move towards 96GG because it's easier to identify them as positive. And I'll let you guys decide whether that's useful or not, because you can talk to some people and they'd say, I'd much rather know if I had CWD. And if you have a herd full of 96GG animals, you'll know pretty quick, because if you put a herd of GG animals in the middle of Iowa County, you'll have it in a couple of years or less. So I'll let you decide whether what's an appropriate thing for you to do for your particular farm. Is this the best we can do? So we have these 4 markers. Like I said, we've identified 3 or more new markers, and we have no idea how resistant they might be. So there's a chance the more we look, the more likely it is we'll find something that does have that extreme level of susceptibility, even if we don't have something with these markers that we know about now. So if the 226K or the 95H isn't it, maybe we'll find something. The more we look, the more we find. Just in the past 12 months, we found 4 new ones that hadn't been reported before. But some of you, and I think I mentioned this the last time I was here, some of you probably have heard about CRISPR for one reason or another, but it's a way, it's a technique that's been used to modify genes in all different species. I think it's even been used in humans at this point. Whether or not that's ethical or legal is another question. But we could completely delete this prion gene from white-tailed deer, and that would make them, they would not be susceptible to CWD at all. And so, this is a question that as an industry, we need to consider whether that's something that we should do. Should we try to develop deer that have this gene knocked out? The deer farming industry would never have to worry about CWD again after that if they did. The concern though is that, if you start talking about this to wildlife professionals or Food and Drug Administration, it becomes a whole new ball of wax where genetically modified organisms, it's okay when we're talking about corn or potatoes, but when we start to talk about a large herbivore like white-tailed deer or even elk, that's a whole nother matter. And genetically modifying large mammals is morally and ethically a little bit more of a concern than genetically modifying corn. So another question, are some of these markers, maybe they're more or less susceptible to different strains of CWD? We don't really know whether that's the case or not. The only thing we have to look at is sheep scraping. and as sheep have been bred to have the ARR marker that I discussed earlier, we haven't seen an increase of scraping in those particular breeds of sheep, despite them having those markers for 5 or 10 years or more now. So it doesn't seem like the scrapie prion is adapting to those animals with those markers. But that's something that we should obviously be concerned about. We don't want to breed 226 KK animals and find out 50 years down the road that now these animals are just as susceptible as the GG animals are. So these are all things that we want to do research on and try to come up with answers so that we can better manage this disease, whether we're talking about farm deer or wild deer. Just kind of a summary and conclusions. Without things like genetic engineering, without things like CRISPR, we're kind of limited to the tools that we have available. So these markers that we've identified, those are the things that we can kind of play with until other markers are identified. We have to kind of focus on what we have available to try to decrease the number of deer that are showing up positive for this disease. We know a fair amount about the different markers and how resistant they might be, but we don't know how this will play out in a herd, in a whole herd population. So that's one of the things that we hope to learn from the study that we're doing with the herd with different prevalence rates to see how much of an impact we have on disease prevalence and ultimately identify whether it's worth it or not. Basically, several of these alleles have the potential to be incredibly resistant to CWD. So the 226K, 95H1, 16G, each of these markers have the potential to be incredibly resistant to infection. Last, we might have to tailor our genetic programs based on what's circulating in that particular area. The depopulations that I showed, again, that's all of North America, Canada, and the US. If there are different strains of CWD, chances are there's a strain that might be different here in Wisconsin than it is in Texas, for example. And so what works for here may not work as well in Texas. And so, we need to keep paying attention to how effective these different alleles are on one property and make changes based on that to kind of improve what we're seeing. So, there's a bunch of people I have to thank. The North American deer farmers have provided funding. Whitetails of Wisconsin have provided funding to do a lot of this research. The deer farmers that have gone through depopulations, some of them have been very helpful at providing samples for genetic testing, but we could always use more information. Some of these really rare alleles we need to learn more about and get more information on. People at the USDA, Tracy Nichols, Aaron Lemkel, and Bruce Thompson, and Justin Greenlee, these are all people at the USDA who have been doing CWD research for a while. I have students that have been working in my lab at the university for several years. Gordon Mitchell with Canadian Food Inspection Agency and Dr. Hohendeltzer here with DACAP, who's been very helpful. So I'm happy to take any questions, and and we'll try to get Dennis up here to talk about testing, sample collection, and that sort of stuff. So the question is, how exactly would the gene-edited animals work? And so this is something that can be done very easily in mice. Mice have a very high reproductive, cycle. They're cycling every month or so, and they're having pups every month or so. It's obviously going to be a lot more difficult with white-tailed deer with only, a single breeding season a year. But ideally, you'd have both a sire and a doe that have at least one knockout, and you try to breed them. And if you have 4 fawns, statistically, one of those is going to be a double knockout. But it's obviously going to be much harder to do that in white-tailed deer than it has been in mice, for example. But yeah, different strategies for trying to make animals that have that gene completely knocked out. Dennis, yeah. Yeah, so that's a good question, right? So this gene obviously has some function. You can't just knock it out and expect nothing bad to happen. All we know is that there are cattle that have had that gene knocked out. Those cattle are completely resistant to mad cow disease, and for the most part, nobody can tell those cows apart from a cow that hasn't had that gene knocked out. So, like I said, this gene is responsible for something. Maybe it makes it easier for the deer to do calculus. I don't know. Do deer really need to do calculus? That's another question. I mean, maybe they do, but farmed cattle that have the gene knocked out appear healthy. Yeah. Good question. So is there anything physically that we can see about animals with these markers that may make it easier to identify them, or that may be associated with some sort of detrimental effect? The answer is no. We've had farmers submit fawns, twins, and the way the breeding works out, you can have twins, one that's an HH and one that's a GG, based on the parentage. And we've had those situations where we've had twins, one's an HH, the other's a GG. The owner wouldn't have known it if we didn't genetic test them. So nothing that we've been able to physically identify so far differentiates markered animals versus non. That's a good question. So the question is basically, we're doing this genetic testing, and sometimes what you'd expect in a fawn doesn't exactly match up based on the parentage with what you actually get for a fawn. So what we've kind of identified by accident over the past year or so is that there are probably versions of this gene that are subtly different, and for one reason or another, every animal has 2 copies of this gene, right? And so what we're hoping is that both of those copies show up as well as the other. But what happens sometimes, and we haven't really been able to explain why, just one copy shows up better than the other. So if you have a if you have a GS animal, for example, it might look like a GG just based on how these alleles are showing up in the test. Conversely, you could have a GS and it looks like an SS. And what we're using is a test that was developed about 15 years ago used exclusively on wild white-tailed deer where they had no idea of parentage, right? And so now that we're using it in farm deer and we know more about parentage, we can see some of the flaws in, maybe there's some difference in the gene in some animals versus the majority of the animals out there that make a GS animal look like a GG or an SS. And so we just have to kind of tweak the test a little bit to try to figure out if we can make it more obvious that that animal is a GG. Yeah, so, and I talked to Dennis about this earlier. Once we've identified animals that we are highly suspicious of having that variability, whatever it is, we can focus on tweaking the test with those animals specifically. Because if we have a GG animal and it looks like it's it's throwing offspring that are all G, can't really look at those animals. We have to look at the animals that we know we're having a problem with, and adjusting the test so